Hello everybody, this is Mr. Lima again, and this is video 2 of our cell division lecture series. This one will be focusing on binary fission in the cell cycle. So before we start talking about the complicated processes of mitosis and meiosis, which are definitely much simpler than anything we've seen before this year so far, the year will get easier from here on. But before we start talking about that, let's talk about the basics. Now, eukaryotic cells, which are more complex, will require complex mechanisms to separate them. But simpler cells, such as prokaryotics, or the original types of life forms on Earth, the ancient life forms on Earth, many of which are still around, replicate through an easier process called binary fission. Basically, all that happens here is the cell splitting in two in a very simple process that works like this. Remember from the previous lecture that the DNA of prokaryotes is basically one singular loop that is attached to the cell membrane. Now, basically what will happen is, like I said in the previous video, prior to any cell division, you have duplication of the DNA. The singular circular-like chromosome will split up, and then the attachment to the membrane will happen on the second chromosome as well, and then that chromosome will separate and attach itself far from wherever the first one was attached, and then the cell will just separate in half in a process called cytokinesis, in which the cell just leaves. And you see that happening here on the top right corner with the bacteria, where it shows the difference between a mother cell and two daughter cells. And you can see them here doing the same thing here, 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 and here. Or even here, or even here. And this is one that just finished probably because they're really close together. Here's another one. So you see, in any bacterial culture, you have thousands of this happening every second, because it's very simple, it's very fast. So that's the major difference between the prokaryotic and eukaryotic cell division, is that binary fission will be much faster than mitosis or meiosis, because it's simple. It doesn't require the complex procedures necessary to separate the nucleus correctly. All right. Also, because there's only one chromosome, you don't need any sort of methods to make sure the proper separation of chromosomes, so that everything is much easier. The control of this is much easier as well. You're going to see in a different video that the control of the eukaryotic cell process of cell division is very complex, but the bacterial cell control is much simpler. It goes through less hoops. And finally, there is, though, a similarity between the two processes, namely the very end, which is called the cytokinesis, when the cyto for cell, kinesis for the division, where the cell actually splits up. So cytokinesis will happen in any kind of cell division, including bacterial and eukaryotic cell division. But that's at the very end. But what happens in between is what's going to be different. I also want to point out before I move on is that there are other kinds of unicellular life that are not bacteria. Unicellular eukaryotes, such as protozoa, algae, and mold. Right? So these things here, for example, you see an example of an algae there and a paramecium down here. Both of them undergo a process that looks very similar to the binary fission we just saw. So here you see a paramecium splitting up. Another example here of protozoa splitting up. And so you see that these processes will be taking place. And you also see in the screen the difference between mitosis, meiosis, and binary fission. Meiosis and mitosis are much more complicated than binary fission is. But I wanted to point out that all eukaryotes, including the unicellular protozoa, algae, and mold, will undergo mitosis prior to actual fission or cytokinesis. True binary fission, which does not require nuclear separation prior to cell separation, only happens in prokaryotic cells called bacteria, basically. And so I wanted to make that clear, because some people get confused the fact that all unicellular life does binary fission, but that's not true. Some unicellular life is eukaryotic. There are simple eukaryotes out there which do mitosis just like you and me before they actually separate in half. So these cells that you're seeing in the board were separated by mitosis, not binary fission. Okay? Now we have to talk about the cell cycle. And the cell cycle is basically the process by which the cell will undergo growth, development, and finally separation. So when an embryo grows from a single zygote all the way down to a fetus, it undergoes thousands of these cell cycles. And that's what we're going to be talking about right now. Now, the cell cycle basically has two major stages, the interphase and then the mitotic phase. And as you can see, the mitotic phase is only a small portion of the cell cycle. In fact, in a 24-hour period, the cell will spend about 20 hours in interphase and only about 4 hours splitting. Okay, and so the interphase can vary between 80 to 90% of the cell life is spent on interphase. Now, some people refer to the interphase as the resting phase, but I don't like that terminology because it makes it look like the cell is doing nothing. It's only resting in the sense that it's not splitting. But on the contrary, during the interphase, the cell is very active. In fact, it's doing everything it normally does. It's doing metabolic activities, making proteins, making amino acids, working with amino acids. It's doing cell respiration, photosynthesis, cell membrane transport, digestion. All of the processes which cells normally do are going to be happening inside these things. It will be doing metabolism, catabolism, and all of those things. Now, the difference is that when you get to the mitotic phase, all of these things are kind of put on halt in prep so that the cell can focus on splitting up. Which means during mitosis, the cell does not do normal metabolic activities. All right? So that's why the cell does this very fast, but it needs to do that in a method to do it efficiently without causing problems. And that's what we're going to be talking about in the next video, which is going to focus on mitosis. Now I just wanted to focus on what actually happens on each of these stages. Now, the interphase, it can actually be split into three parts, okay? Well, remember that all of the three parts, the cell will be doing normal metabolic activities. But each of these three phases will also, at the same time of metabolic activities, something will be doing in reference to the actual division. Because although it's called a resting phase or non-dividing phase, these cells will be doing something that has to do with dividing, either after division or in preparation for division. So basically, right after division completes, the cell starts a growth phase where it goes from the small daughter cell and it grows to a normal-sized adult cell. And that will include the duplication of the organelles, synthesis of a lot of proteins, in addition to all the normal metabolic activities that cells will have to undergo. All right? Now, after the initial growth phase, the cell will hit a point where it will grow too large, like we talked about in the previous video. As it starts getting close to this point, it gathers a bunch of chemicals inside the cells, which basically send a signal for the cell it's time to divide. And the very first thing that happens in response to this chemical signal is that the DNA of the cell is replicated. So the chromosomes go from that singular chromosomal structure, which actually it's in chromatin shape, but the chromatin starts to duplicate in that single linear DNA called molecule will double itself up to become the familiar X-shaped chromosome with two sister chromatids held together by a centromere, right? And then it will have on the top the short arm and the long arm, like we talked about in the previous video. And then at the end of the chromosomes, you have sequences called telomeres, which protect the chromosome from degradation. Now, this basic structure of the chromosome only becomes visible prior to the actual mitotic phase down here. During this part right here, it's all it is double the chromatin. So you won't see the actual called up chromosome until right before mitosis actually happens. Okay? Remember that it's during the synthesis phase, also called S phase, that this is happening. So you see the S phase there. Now, then the cell will go into a second growth phase called the G2 phase. Remember that this cell is going to split in half. You don't want the daughter cells to be too small. So prior to the actual division, the cell will grow excessively large. Too large, in fact, to contain itself. We talked about this. The cell, when it gets too large, it becomes problematic, the process. But the cell briefly grows to that point where it becomes too large because it knows that it's going to have to split up in half, and it knows that each half will have to be decently sized. You know, yes, each half is going to grow again through the G1 phase, but you don't want each half to be too small for the cell to have problems. So fitting everything it needs to fit in there. So the G2 phase will make sure that the cell is large enough. Other things will be happening during the G2 phase itself. Also, energy for the mitosis process is going to start being produced and stored. A lot of ATP production, getting ready to be used during the mitotic phase. Also, the DNA is going to be checked for errors to make sure that nothing's wrong with the DNA following the synthesis phase. Also, you're going to need to duplicate all the organelles. You're going to need to make more cytoskeleton because the cytoskeleton is going to be used during the actual division process. Also, you're going to need to prepare chemically for the cell division process. So all of these changes will be happening in the cell during the G2 phase. But remember that during the G2 phase and the S phase, the cell will continue to do normal metabolic activities. But then eventually, these chemicals which are sending the message for the cell to divide will gather so much inside the cell that at the end of the G2 phase, enough is enough and the cell says it's time to divide, and then the cell will undergo the mitotic process, upon which it will split up. And this mitotic process takes 4 phases: prophase, metaphase, anaphase, and telophase. And then at the very end of telophase, the cell actually splits into a process similar to binary fission, which is called cytokinesis, and that's pretty much how it works. Now, before I finish up the video, notice the DNA count in the cell. At the end of G1, you have the normal amount of DNA from the cell. You notice that there's only about a normal amount of DNA. But at the end of G1, the cell will start doubling the DNA and becoming doubled. You see that here. And that double DNA will proceed throughout G2 until it splits in half and the cell again returns to half the DNA because each of the daughter cells get 1/2. Also notice this G0 business here. Cells called G0 go into what's called a cell cycle arrest. Now these are cells which cannot continue mitosis. They can never replicate. They can never grow and never duplicate. Why would you need a cell that can't do that? Remember, think about it. We talked about it in the previous video. You need to grow in your body. You need replication because cells have a limited size. You need replication for fixing problems, for tissue repair. And you need replication for reproduction purposes. So what kind of cell would not want to reproduce? I mean, that doesn't seem to make sense. Well, there are some cells which can reproduce. An example of these cells are neurons and muscle cells. You see, these cells, the whole premise of nerve cells is that they are specific connections between these nerve cells. If you were to duplicate one of them, you would automatically have to break these connections in order to make 2 new cells, which would then have to make 2 new connections. But since memory and control is all about how each neuron connects to another, you can't duplicate because it would actually break the connections between your neurons. Not only that, these muscle cells and nerve cells can't stop working. They're constantly, for example, how is the diaphragm cell, the heart muscle cell, the nerve cells, which are constantly doing their jobs. They can never afford to go through a mitotic stage where the metabolic activities stop. Think about what would happen to your brain functioning if your cells decided to just stop working for a bit. Even if it's 1 or 2 here and there. The body cannot afford to shut down nerve impulses. It cannot afford to break and rebuild connections. And remember, the nerve cells go from the spinal cord all the end to the fingertips of your fingers. And so, if you have to duplicate a nerve cell, you would definitely have to duplicate that entire ginormous cell. That's just not going to happen. Which means, once the nerve cells are produced, they were going to stay the way they are for the rest of their lives. And they will never, ever progress past that point. And which means it causes problems for the nervous system or muscle cells. Because these cells can't repair themselves. The nervous system cannot repair itself. Damage to the nervous systems and muscles is serious. For example, when you have a cardiac muscle death because of a heart attack, or you win a stroke that causes problems in the brain, that tissue will never recover because it doesn't naturally replicate itself unless somehow science induces that process to happen. So these are G0 cells, and they are important within the anatomy, and I hope you understand what the cell cycle is all about and how it works. On the next video, we're going to be talking about that small 10% piece called mitosis. See you then.